Good evening, Apurba sir. Good evening, sir. Good evening, sir.
Good evening, sir. Hello.
There is a problem.
Hello.
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 Yes, sir, you're audible.
Okay, am I audible? Okay. I'll have to share my screen with the slides, right? Okay.
Yes, sir.
So what it is popping up here, I think. Let me close this one. And then. What is this share button? I think it is on the top, right? Somewhere or
No,
on the left?
Below.
Oh, below. On the left?
Yes, below right, after webcam.
Oh, acha. This is
Share screen, then application window, this way.
application window. Below on the right.
Right.
Okay.
Below right.
But that says log out. Oh,
No, before that. After share webcam.
select slides.
Select slides here. This one.
Oh, select slides, right? So I can pick up. No, but this is not the one. Okay, let me see. That day I could have done that. I mean that day I did it very.
Okay, actually initially you share a whole of your screen. I think you have to stop sharing option. Then after that again you start the sharing.
Okay, so
Because initially it is showing your whole screen. We already see your whole screen.
oh, you can see my whole screen.
But after that it is not coming. Initially we
Notes.
see your whole screen.
Just a moment. I think if I can drag that, will that work?
No.
No. If I drag my file, will that is it going to work? No.
No.
Sir, I am telling, sir.
Yeah.
I am telling, sir. Please, first of all, open your sir PPT. Whatever you want, and that please open that one.
Okay. So at this moment, can I log out?
No need to log out, sir.
Okay.
You minimize the browser.
I can open my slides, right?
Yes,
I'm
yes.
opening. Yeah, I could. Just a second. Yeah, I've opened that.
Yes, sir. Now, sir, on that screen, click on share screen. Beside webcam button. Beside webcam button.
Webcam is here. Share screen. Okay.
Yes,
Share screen. I think. Yeah.
Then sir, there are three options. Are there entire screen, application window. Click on application window. At the top.
Application window. Okay. Now. Where is the application window?
Are you getting the entire screen, application window, and
No, I'm getting on the left the messages, and then the user and participants, all these things, and then I see some names here, but not any application window for say. No. application window.
Sir, sir, which browser you are using?
I'm using my computer browser, but I mean, of course, it is.
Okay. Sir, your full screen is visible. Your full screen is visible. Now open your PPT file, then we can see.
Yeah, this is my full. Yeah, so I think you can see now, right?
Yes.
Now,
Yes.
can you see that my PPT
Yes, sir.
files?
Yes, sir.
Okay.
Please put
Yeah.
in the show mode so we can view the entire.
Okay. What is this? Another thing is popping up here, which is, what is this? There is, the whole screen is, you know.
Yes, sir. Please click on your PPT file.
PPT file is here. Yeah.
Yes, sir.
Yeah,
Then,
this is my PPT file. Oh, it is going out, you know. See, this is not allowing me to go. Anyway, what is this? This session is. No. See, that day you gave me an option to, you know, upload the files. So where is that? Then I can.
yes, that you can do that at the bottom left side, the plus sign is there.
Yes.
So click on that.
Upload a presentation. Okay.
Yes, sir.
And then I can.
Then browse the file.
Yeah.
Click on that.
Yeah.
Then choose your file.
Yeah. That is exactly, I did it last time also. Yeah. Choose the for upload.
Yes, sir.
Yeah. It is coming up.
Wait little. Yes.
Yeah.
Then click on after. So it should.
After it uploads, yeah, I can. I'll do that. There is another option also. The drag the files
Yes.
here to upload.
Yes.
I think what is happening? To be uploaded, it says.
Sir,
Should I drag it?
No.
Can
Sir,
I drag?
Click on upload at the top. Orange
Yeah.
button.
Okay. Yeah.
Upload, you just put it, click on here, it will automatically upload.
Just wait, sir.
Yeah, it is uploading.
Uploading. Yes, sir. Yes, sir.
But you can see me, right? Do you?
Yes, sir. Yes, sir.
Okay, no problem. Then let it upload and I can start, you know, discussing of whatever we have in the slides.
Tanushree ma'am. Ma'am,
Hello?
there is doing uploading, you just start the session.
Yes, sir.
Okay.
So, good afternoon, everyone. So, we have come to the second last session of the second day FDP, online FDP. This session will be taken by Professor Professor Apurba Bhattacharjee. He's from Georgetown University, Washington, USA. He will be talking on pharmacophore modeling and its usefulness in the discovery and design of potential drug molecules. Dr. Apurba Bhattacharjee is currently an adjunct faculty professor at the Department of Microbiology and Immunology at the School of Medicine, Georgetown University Medical Center, that is Georgetown University, Washington, D.C., USA. Earlier, he was the chief molecular modeler, that is the senior computational chemistry scientist at the Walter Reed Army Institute of Research, Maryland, USA. He has over 30 years of research experience in computer-aided drug design and discovery, particularly in the applications of quantum chemistry and pharmacophore modeling in delivery of anti-malarial, anti-leishmanial, insects, repellents, and counter-door field of discovery of antivirals targeting specifically the dengue, Zika, and more recently the SARS-CoV-2, that is COVID-19 virus. He has authored and co-authored five book chapters and 5 papers. Now I will request section.
Do you hear me? Good morning and good afternoon in India. Here it is morning at 8:30. So first of all, I thank the organizer for giving me this opportunity to share some of my research and our group research on pharmacophore modeling and its usefulness, the discovery and design of potential drug molecules. So in today's talk, I plan to this is the way. Today's talk, I plan to outline these followings: a brief introduction to drug discovery and development, the emergence and necessity of the in silico methods. Dr. Mitra, can you hear me? And is it okay?
Yes. I hear you, sir. I hear you.
Okay, thank you. Because the screen, I have made it the full screen so that I can work on it better than the other way. Okay. So the outlines will be these followings: emergence of and the necessity of in silico methods, their importance and usefulness, and my focus would be particularly on the pharmacophore modeling, and I'll present two case studies from our own research, where the target protein structure is known in one case, and the other case the target protein structure of the disease is unknown. So, excuse me. My special focus would be on pharmacophore modeling. Pharmacophore modeling is a kind of weak interactions with the drug molecules, the active site of the drug molecules, and this has got immense implication in material design, the cluster design, the molecular cluster design, which are very commonly used these days for the material sciences. The next slide, I would like to, mention that the drug discovery and development is a complex and a tedious process, in the sense that it takes almost 10 to 12 years for a new drug molecule from the bench of discovery to the shelf of a pharmacy. So obviously it is a very lengthy process, and more important that it cost a staggering 3 to 5 billion US dollars, and only 1/3 of this cost actually goes for the discovery. The most of the money is spent for development. Now, development is the process which is where most of the drug molecules fail, and attrition occurs because of the bioavailability, toxicity, and various other problems associated with the drug. So phase 1, phase 2, and phase 3, these are the 3 different clinical trials where most of the money is spent. And only 3 out of 10 drugs achieve the revenues which are greater than the development cost. So you can imagine that is a very expensive matter. So obviously there is a necessity to minimize the cost, and in silico methods emerged in this way for the last 30 years or so. Now let's see that what are the different stages of discovery and development of a drug. See, the first thing what we need to do is to identify the disease, and then we'll have to isolate the protein involved in the disease. This part of the study usually has to do with the genome genomics and the proteomics. And once we know the active site of the protein structure, if most often they are carried out with X-ray crystallography and other means. Electron microscopy also sometimes help in identifying the real protein structure involved in the disease. Now, once we get the structure, then we can look for compounds which can effectively bind to this and inhibit the biological response of the disease. Now, this is primarily the task of the drug discovery, but beyond that, one has to prove the validity of a molecule by in vitro studies first, and if shows promise, then go for the animal studies. Beyond animal studies, we'll have to find out the formulation of the drug molecule, how it is going to be delivered, and once we know all these things, we can scale up the process, and then we can find an IND. IND is an investigational new drug. And after this stage, the actual development process starts, where the clinical trials, human clinical trials, all these things. And you have seen in the recent times that how long it took for phase 1, phase 2. Because, the discovery was done way back for the COVID-19 vaccine, perhaps in the early May, or even by most of the companies in the world, they could find out the vaccine. Discovery of the vaccine by May, but the June onwards, it was totally the trials, the actual trial, and then it takes about 2 to 10 years in this process usually, and then then finally you can file a new drug application, which again is to be approved by the FDA, Food and Drug Administration in the United States, before it can reach the shelf of a pharmacy. So it's a lengthy and very tedious process, undoubtedly. So, and most of the money is spent on this development of the drug molecule. Now the problem is for a new drug molecule, since it takes such a long time and it costs so much, the in silico methods, they can help in reducing not only the cost, but also the time by quickly, shortlisting compounds which should be available in the regular databases and things like that. So, but let us try to get some background of the whole thing. It is in silico methods are nothing but the computer science application of computer science to solve chemical and biological problems. Now you can create algorithms and also softwares to visualize the structure and the properties of the molecules. And computing power, the astronomical computer power these days have really has changed the idea from the early 1990s to the present time, that it is a huge power for the computers to streamline the processes. Now we can leverage the chemical and biological information about the ligands, the targets, to identify and optimize the drugs. Then again, another thing that in the development phase, most of the failures occur because of the ADME properties. ADME is nothing but absorption, distribution, metabolism, excretion, and toxicity. These are together called ADMET properties. So those are the filters can be calculated using in silico techniques. So they can also solve a lot of problems and minimize the time involved in the process. Now, virtual screening, this is one technique, is an in silico technique, which is commonly used these days. These days means at least for the last 10 years or so, where you can, using this in silico methods, you can shortlist the number of compounds for testing, the actual testing, that means the experimental testing. So what is the process of the virtual screening? We need to have know some three-dimensional structure of the target, that means disease-specific target, that is the protein structure. And once we know that, then the life becomes a lot simpler. that you can use the molecules by from the databases, the compound databases, some different sources, and dock these molecules in the active site, as you see here that I've shown on the right-hand side of the screen. These are called the structure-based methods. So what is done that we quickly calculate the binding energies of hundreds and thousands of compounds and see that what are the minimum binding energies, and then shortlist the compounds based on certain characteristics of drug likeness, and then you can test them finally in the in vitro and in vivo system. Now see, the interesting part here that these molecules which bind to the active site of the protein, they are to interact with the protein active site in the form of weak interactions. Because if it is a covalent interaction, then the molecule is going to sit there forever, which is never wanted. It is definitely not the favorable approach for drug discovery. So we'll have to see that the molecules will have to kind of interact with the active site with a weak interaction. Weak interactions will mean that hydrogen bond interactions, then hydrophobic interactions, like van der Waals interactions, pi-pi interaction, cation-pi interaction, etc. Now, this is one approach where by docking we can find out and shortlist the number of compounds. The other approach is where we don't know the protein structure. And that happens very often because, many of the diseases, we know the disease and we know some compounds which are active against the disease or can inhibit the biological response of the disease.
The program வந்து டெல்லியில இருந்து நடத்துறாங்க.
Excuse me? Hello? Dr. Mitra?
No, sir. Continue, sir.
No problem. Please continue, sir.
Oh, I'm sorry. I thought that somebody is asking me some question.
No, it is a mistake.
Okay. So the other approach is where you don't know the target structure. Then what happens, and this is very common, that there are many diseases where the targets are yet unknown. Like even for malaria, we don't know for sure that which is the target molecule. Now, in that cases, we'll have to use the ligand-based approaches. Ligand-based approaches means, knowing some active compounds. That means there may be, like, although malarial, the protein structure of the disease is not known, but we know that there are so many compounds available which are active against malaria. So based on the characteristics of the structural characteristics, the chemical structural characteristics, one can, find out completely new class of compounds which are less toxic and easily bioavailable, and all other things using in silico techniques. So there are two approaches common here. The actives is similarity searching, and the other one is pharmacophore mapping and modeling. And this is the thing which I will focus mostly today. And there is another thing also, the actives and inactives, if both are known, then we can do a machine learning, that means artificial intelligence techniques to find out new compounds. Here, actually, at this the current moment, we are working in intelligent augmentation of these machine learning tools using Pharmacophore. That means Pharmacophore can help the process of down selecting compounds using machine learning tools. Anyway, next is, these are the filters. You can, these are in silico filters. You can quickly calculate them, and they are very simple. And this is the reference behind this, that if you have to have a molecular weight less than 500, a log P less than 5, hydrogen bond numbers should not be greater than 5. It should be always less than 5, less than 10, the hydrogen bond acceptors. And then the rotatable bonds are less than 10. The polar surface area, this is an important characteristics for hydrophobicity in a molecule, but that should also be less than 140 square angstrom. And another thing important is that the peptides are usually unsuitable for for the small molecule drugs. Yeah. However, all this looks very simple, that the virtual screening can quickly overcome a lot of these hurdles, but it is not really that simple. So there are quite a few challenges in the virtual screening. See, it is estimated that about 1 billion compounds, drug-like compounds, are there in the chemical space, and about 10 to the power 4 to 5 human proteins that are known in the biological space. So now, to choose the right molecule for the right target is not a very easy thing. It takes a huge, patience, and also huge, I would say, the time also to select the right way. So what happens, another problem is that although we see that about 1 billion compounds are there in the chemical space, actually only 30 million are available compounds in databases. So, and then, a large pharmaceutical company can at best handle 1 million compound in its HTS, that is the high throughput screening. What is done that, all the compounds you choose, and they have to be put in the high throughput screening for quick screening of their activity. And that is also a very complicated process. It usually is a robotic way of carrying it out, but it takes a long time also for 30 million to be achieved. So it is not a joke. And another thing, that all targets are not the valid targets. This is another important thing. So if the targets, although we know that there are only 10 to the power 4 and to 5, yet that number is a very big one. But, for a computer, it can quickly search it. But it does not mean that, all targets are validated targets. So, and another thing also, the, large-scale screening is very expensive. So, usually, these are the few challenges the in silico virtual screening takes, to address to. And they're working on various techniques. Your machine learning techniques, one which is very commonly now used. Okay, now let's see that I'm going back to pharmacophore modeling and see that how what are the basic concept of pharmacophore, and then we'll see that how it can be very quickly applied for down selection of the compounds. Now, a molecule, we know that when a molecule can be a drug molecule. So if we have the target structure of the disease-specific target, then we know that when the molecule can optimally interact with the target structure to trigger or inhibit its biological response, then the molecule has the potential to be a drug molecule. This is as simple as that. But in other case, what happens that we know there are some compounds which work against a drug molecule. And, we obviously understand that since the molecule is working against that disease, and the molecule must be optimally interacting with the disease-specific structure to trigger the biological response or inhibit the biological response. Now, at the molecular level, what we mean by a molecule? The molecule has two very important characteristics. That is the steric characteristics, steric part, and the electronic characteristics and electronic features. That is, so we can say that this is nothing but a combination of steric and electronic. We can combine and say that stereoelectronic features, which are responsible for optimally binding with the specific biological target structure to trigger the biological response. And this is the idea behind the pharmacophore. So pharmacophore essentially is nothing but a molecule, the stereoelectronic features of a molecule which can interact with the active site of the disease-specific biological target to trigger its biological response or inhibit its biological response, either way. The next slide, oh, what happened? Yeah, this is the definition provided by IUPAC about the pharmacophore. It is in 1998. Yeah, it is an ensemble of steric and electronic features necessary for optimal interaction with a specific biological target structure to trigger or inhibit its biological response. And this was published in 1998. So this is still the valid structure. There are certain modifications made over the years. I will see that later on, how we can apply this. And that is the next stage. I'll draw your attention in few of this, particular on this slide, to explain the point of pharmacophore. You see, this is a two-dimensional representation of a molecule. This is called mefloquine, and mefloquine is known here as Lariam. It's an antimalarial drug. So this is the two-dimensional two-dimensional depiction of this molecule. Now, if you minimize this structure, that means in a computer we can develop these kinds of different things, the tube structure, then skeleton structures, and etc. But what is more important that if you come to these two. Now, this is the space filling. That means the van der Waals surface of the atoms together. And this is the electron density. This is the molecular electron density, and this is the electrostatic potential. I will draw your attention to these two specific figures to explain the point on pharmacophore. Now, electron density and electrostatic potential surface both can be experimentally determined by X-ray diffraction patterns. Now, electron density, how it arises? It is because of the nuclear distribution and the electron distribution due to the presence of different atoms in a molecule. So they redistribute in the molecule in a form that certain regions of the molecule may have a higher density of electrons, whereas certain regions of the molecule will be deficient of electrons. Now, this can be very clearly seen here, and we can calculate this one using quantum mechanics. See, this part is the nuclear charge distribution, and this part is the electron charge distribution in the equation, and this can be combined, and Hartree-Fock or even density functional theory can quite accurately calculate this, and also can be determined using X-ray diffraction patterns. Now, here, I try to explain this part a little bit, that when we bring a positive charge near this electron density surface, the positive charge, just imagine a hydrogen ion brought to the proximity of this molecule, then the hydrogen ion, or just think about a proton, will experience a different kind of interaction in different regions of the molecule. So the regions where there are large electron density will experience, the proton will experience a very strong attractive interaction. And that interaction in potential term is called the most negative potential region. And conversely, the area which is most deficient in electrons, that will experience a kind of a repulsive interaction, and that is called in electrostatic potential terms the most positive potential. So in this way, you can identify two extreme regions in the molecule where we have the maximum density of electrons and where we have the least density of the electrons. And these are depicted with two extreme colors. The red color would indicate the most negative potential region in the molecule, and the most deepest blue color will indicate the most positive potential in the region. Now, what are the implications of this? The most negative potential region in the molecule, obviously, since it have the highest electron density, will also function as a nucleophilic region in the molecule, or we can consider that as a hydrogen bond acceptor region, because hydrogen bonds can easily be formed with that part of the molecule where it has got the largest electron density. On the other hand, when there is the least electron density, this can function as a hydrogen bond donor. So what happens there, that the hydrogen bonds, because of the positive potential, the other side can accept a hydrogen bond, the target structure. There is a complementary site in the target structure which can easily accept that hydrogen bond donor site, and the other side is a hydrogen bond acceptor site. And the colors, the intermediate shades of color, they are called the weak electrostatic potential region, and which can be considered as the hydrophobic region because they are very weak. It's like, regular pi or, the non-bonded other kinds of interactions, which are very common in the protein structures. Now, so what we get out of this, that there are three distinct features we can identify. They are all electronic features. That there is a hydrogen bond donor site, a hydrogen bond donor site, the deepest blue region, the deepest red region is the hydrogen bond acceptor site, and there is a large area of hydrophobic nature in the molecule. And there is another also, so we see that there are two hydrogen bond acceptor sites in this molecule. There is one very distinctly hydrogen bond donor site and a large hydrophobic site. So these are the main electronic features we identify from this molecule, and this is called the interaction pharmacophore of this molecule. Now, another interesting thing which I should also mention, this is the steric thing. Now, out of these two hydrogen bond acceptor here, you see that this nitrogen atom is a hydrogen bond acceptor, like this one, this nitrogen atom. If you go back to the two-dimensional structure, you can see that this nitrogen is the hydrogen bond acceptor, and this nitrogen is also another hydrogen bond acceptor. But you see, out of these two, this nitrogen is sterically hindered by these two big CF3, that is the trifluorocarbon groups here. You see that. So what happens that this will be, although accessible, but it is more sterically hindered. Now, as a result, what will happen that this nitrogen atom will be more easily accessible to the active site of the protein molecule. So this together, we can obviously consider now, these are the stereoelectronic features associated with mefloquine molecule, and these features will be recognized by the active site of the protein structure to promote interaction at a distance. And once it recognizes this, it will come closer to each other, and then the binding will take place. And that is why it is called the interaction pharmacophore. And this is the foundation of the pharmacophore modeling. Oh, what happened? All right. Now you see, I'll go a little further with this idea that, and how it can be useful for quickly identifying new class of compounds. You see here, this was a part of the work which we did in Walter Reed, that the antibacterial thiolactomycin, this is the chemical structure, the two-dimensional chemical structure of this one. This was the only known compound against FABH. FABH is a fatty acid biosynthetic enzyme, which are very useful to target for malaria disease. So we are trying to find out new class of compounds because this was not that good, neither in inhibition nor as an active antimalarial. So first we did the van der Waals surface, that means the electrostatic potential on the molecular surface, and this is how the electrostatic potential profile looks like. And if you go beyond this surface, this is interesting, that van der Waals surface and beyond van der Waals surface, that is about 1.3 angstrom away from the van der Waals surface, the potential looks somewhat like this. And then if you go even farther, like 1.4 angstrom away, we see a large extended region forming in the molecule. And even beyond that, like at a 1.5 angstrom away, we see a real large, this is called the nucleophilic suction-like appearance in the molecule. Now, this is a high and powerful recognition feature of this molecule. Now, this part of the molecule can easily be recognized by the protein structure at a distance. So what happened that we took a very probabilistic idea of making a simple pharmacophore of 2 hydrogen bond donors at one side of the molecule. And since this part of the molecule has a very weak electrostatic field, we put that hydrophobic site here. And together with this shape, that means the three-dimensional shape of the molecule, we made a template of this molecule, thiolactamycin, and used this for a database search. And then it gave us very quickly, within a short time, a very important compound which has the inhibition of the FABH enzyme, 82%. And this is the structure of the molecule. This is no way same as thiolactamycin. And this molecule is also a very, very good antimalarial compound. And this was been done, and we published this paper also way back in 2009. So you can see the usefulness of the pharmacophore model and electrostatic potential interaction pharmacophore to get a very quick view and useful identification of a new compound. The next one, so pharmacophore, although it is an abstract concept, yet it can be utilized for, with certain features, for example, the hydrogen bond, the hydrophobic ring aromaticity, ionizable sites, etc. You see, the ring aromaticity is an important thing that it can function in a protein structure, both as a cation pi interactions and also as an anion pi interaction. The hydrogen, they are all weak interactions, remember. The ionizable sites also certain times can function as a very weak interaction, but many a time it can function also as a very strong electrostatic bonds type of function. Anyway, so we can put certain figures like hydrogen bond acceptors and hydrogen bond donors, hydrophobic site with the different colors in the molecule, and use that as a template. This is the statistical approach where the databases can quickly recognize these features and down select the compounds. The next, so the essential idea is that if a pharmacophore model shares two dissimilar molecules with the one of them, the activity of one of them, if it's known, the other one can be predicted very quickly. For example, here we see that these two dissimilar molecules, they share the same pharmacophore. These are the two hydrogen bond acceptors in both of them and two hydrophobic sites. So since this molecule is experimentally found to be antimalarial, we predicted that this molecule, since that shares the same pharmacophore model, it should also be antimalarial. And indeed, we found that this is also highly potent antimalarial. So methods used, we start with the semi-empirical and ab initio quantum mechanics, mostly in these calculations. And then there are certain statistical methods like, Catalyst, Discovery Studio, Comfort. These are very commonly used these days. And of course, we'll have to make the database ready for, using this kind of pharmacophore for quick down selection of compounds. They are to be made in a 3D multi-conformer format. Then, of course, this is the most important thing, that even after down selecting the compounds, unless we test them, there is no validity of the pharmacophore model. So now let me go through certain specific case studies which we performed in Walter Reed. I'm not going to talk about COVID-19 at this moment because, we have done some work but that is part of the university, obligation, and we cannot publish, neither we can publish nor we can discuss about those things unless university permits us. Anyway, so these are the two things, two case studies I will put forward to you and see that how we can use the pharmacophore modeling for discovery of new class of compound. So the first one, it is the discovery of non-oxime reactivators for DFP. DFP is diisopropyl fluorophosphate. This is an organophosphorus compound, which I don't know whether you are familiar with that. Organophosphorus compounds are very, very known poisoning agents and usually used in the warfare. Some of the countries have used them even in recent times, like in Syria, there was a war, the ISIS, and they were using this kind of organophosphorus compounds. But anyway, what is to be done that, we know that organic phosphorus compounds, if they are used in warfare, there are certain compounds called oximes, which can quickly reactivate the system. I'll explain a little more about this one, the background of the whole idea. You see, in the brain, we have something called the acetylcholinesterase. This is an enzyme which is very important to prevent seizures in the brain. What it does, it takes away the acetylcholine. You heard about the acetylcholine. Acetylcholine is a neurotransmitter in the synaptic junction of the nerve terminals. So what happens that the acetylcholine will be continuously hydrolyzed by this particular enzyme. Now, when organophosphorus compounds, the poisoning agents, they interact with a person, the acetylcholinesterase is immediately inhibited. So as a result, what happens, the acetylcholine starts building up in the brain, and that causes seizure. And if that seizure starts, if it is not treated, it could be fatal. So there are the common nerve agents which are used in warfare, is sarin, soman, taboon, and DFP. And there are news, Russia uses this one, the cyclosarin. And the pesticides also. Pesticides are also, they are also capable of inhibiting acetylcholinesterase. And the known oxime reactors, which is used by United States, is 2-PAM. This is I'll show you the structure. This is an oxime. Oximes have several issues, like, it does not go to the brain, which is very important at this moment for treatment. And because, unless the drug goes to the brain, it cannot function to take away the organophosphorus compound from the brain, from the ACHE, that means the acetylcholinesterase. And, it does, but it does it very slow process, in a very slow process. So this is the structure of the acetylcholinesterase. And you see, this is the active site of this molecule. It's a 20 angstrom gauge here. So the compound, the oxime, will have to enter through this and then knock out the organophosphorus compound. So what were the oximes known? These are the oximes, all are, they are known compound, and most of the countries, they use this kind of things except US. US does only a smaller one, 2-PAM. And we have optimized their chemical structures using high level of quantum chemical theory. And you see that these are the structures after that. And we calculated the electrostatic potential of them. And he found something very interesting, that, there is a large electrostatic, the weak force electrostatic potential over the molecule. There are very distinct negative potential region in all of them. And, the nucleophilicity of the oxime hydrogen, oxygen is an important idea from here we got. And the specific location of the molecular orbitals here, that is also can be seen very easily from that, and this also makes sense for electron transfer. Then you see here, ultimately using those calculations, we developed a model, and the model was very simple: one hydrogen bond acceptor, one hydrogen bond donor, and an aromatic hydrophobicity. This was the template of the Pharmacophore, and this was published in way back in 2010. So this particular template of the Pharmacophore allowed, first of all, we check the validity of the Pharmacophore. We see that this Pharmacophore maps on very good compounds like obidoxime, trimodoxime, and there are some key 027. These compounds are mostly Czech Republic compounds and also some Russian compounds. And the crystallographic observations really support the Pharmacophore. That means these are the features necessary for binding to the active site. Next, we use this for database searches. We perform 3 database searches. These are the common commercial databases, Nabreach, Chem Navigator, and we also had one in-house database. These are, of course, proprietary databases. And then we found, very interestingly, some 11 compounds. Yeah, we down selected them, through docking experiments, and then also looking at the blood-brain barrier. These are all in silico studies. Rat oral LD50s, all these things can be calculated. Then finally we got these compounds. See, they are not oximes at all. They are quite different. And I'll show you 11 compounds, no, 12 compounds, I'm sorry. 12 compounds were identified and shortlisted for both in vitro and animal studies. In vitro studies also, they performed very well. And so we select, their binding energies. You see, I mentioned about that while you dock the compound, you'll have to calculate the binding energies. So then what we did, that out of this 12 compounds, 5 were selected for animal studies. Animal studies, I don't know why it is. Animal studies, how do I make it? Oh. Sorry. You see, out of these 12 compounds, 5 were selected based on in vitro activity. The 5 were selected for animal studies. Our animal model was a guinea pig model. And then, we used this 5 compounds separately with the control of 2-PAM, because 2-PAM is the one which is an oxime which is used by the United States Army to get rid of the organophosphorus compound. So we used non-oximes. Those non-oximes, out of those 5, 3 could not, make the animals survive for more than 24 hours. But the 2 of them, number 6 and number 7, they were very useful in the sense that they not only allowed them, the guinea pigs to survive more than 24 hours, but also the guinea pigs were looking very normal after administration of this. So what is done there, the guinea pigs are first given the DFP, that organophosphorus compounds, and when they started having the seizures, we used these compounds to treat them. And 2 of them made the guinea pigs survive not only 24 hours, but also they looked very natural after that time. Now, if you look at then with the histopathology studies, that means the brain, we look at the hippocampus, And this is the electron density, no, sorry, electron density, I'm sorry, electron microscopic picture, the photograph of that. This is the 2PAM, the control. This is the oximes, and these are the non-oximes. You can see that the non-oxime did fairly well with in compared to 2PAM. Although 7 shows a little bit of dysfunction, but yet, 7 was also very good otherwise. The next slide will show that this, so you can see that this is 2PAM, this is 7 and 6. They function even better than 2PAM. So this was the stage that means, from a pharmacophore model, you can go all the way to animal studies and find out compounds which should be really useful for development of a drug. Yeah, so the summary of the work that, we not only found out non-oxime reactivators, but also we showed that the in vitro efficacy and in vivo efficacy of these compounds, which is comparable to 2PAM against the brain symptoms of DFT-induced neuropathology. And the crystallographic observations also supported our pharmacophore. Oh, what happened? I think I'm clicking in a bad place. Okay. See here, this is the other case study, the unknown target structure. Here what we did, that the structure of the protein, the disease-specific target was not known, but the compounds are highly active against a variety of pathogens. These are called the indoloquinazole diones. They were extracted in excess tryptophan. That is why also they are known as tryptanthines. And this was originally, in the early 60s, was extracted from an alkaloid, isolated from a Taiwanese plant, Strobilanthes gusia. But now it is found in many countries and even in the United States, this plant, and we can work from here. So what is important here, that although there are some reports about the antimalarial activity of this compound, but this was not known to Walter Reed. So Walter Reed, Army Institute of Research, my alma mater, the previous company where I worked, they revisited the compounds in 1990s and found, surprisingly, that 150 of these compounds were extremely active. That means, they are so highly potent, less than 100 nanogram per ml, that, you cannot imagine that not a single compound was known as an antimalarial with this kind of potency, the activity. And, not only that, some of these compounds were active against chloroquine, which is 40% resistance already. And the mefloquine, the compound which I have shown you earlier, that is 20% resistance. So this particular compound can work against them so well that it has brought a euphoria, and it was such a surprising discovery that we thought that the malaria problem is solved forever. But unfortunately, this euphoria did not last long. What happened that these molecules, these compounds were practically like hard rocks. They are not bioavailable at all. It cannot be made to work in a human body. But I hope I missed any slide. No. Anyway, so this was not bioavailable in the body. So that is why neither in the animals nor in the humans it could work. And we had to, the army discarded these compounds in the early 2000s. But interesting thing is that since the molecule, these tryptanthines have a very powerful template of activity, that means the pharmacophore. So what we thought that why not build up the pharmacophore model and use that model to search the databases to find out a completely new class of compounds. And indeed we performed that and we are successful. That's what I'm going to show you. So we did some training set of these known compounds with this about 17 of them. And then we Yeah, we test them also in another test set that whether the pharmacophore works or not. And then this was our pharmacophore model. Oh, this is the correlation between the activity, the predicted activity and the actual activity. And you see that the correlations are not bad. So then, yeah, this is the pharmacophore model. I think I mentioned to you earlier, this I've shown you this picture. This is the 2 hydrogen bond acceptors flanked by 2 aromatic hydrophobic features. Now this model, what we did next, sorry. Yeah, this model was validated against the tryptanthanes. This is the structure, and then, it was also validated by mapping on various other known antimalarials like quinine, nefloquine, primaquine, chloroquine, etc., hydroxychloroquine, many other compounds. Some of them they map very well, some of them don't map. So we know that their function, the mechanism of actions will be different from the one where tryptantane works. So then, what we did, that we put that pharmacophore model on a very, very potent and very active tryptantane molecule, and then made a shape-based template and searched the database, the in-house database first. And we found that there are quite a few compounds, well, after the ADME toxicity in silico evaluations, the favorable compounds were already selected. We found, surprisingly, 4 different class of compounds, totally different from tryptanthanes, and all of them were found to be excellent antimalarial compound. So this is the, I would say that this is the beauty of pharmacophore model, that you don't have to go for a single category of compounds. You can utilize the model to search the database to find out completely new class of compounds. So pharmacophore, I mean, these are the few concluding remarks I would like to make. The pharmacophore models are very useful and efficient. And the important to note that there is no guarantee that the compounds identified by pharmacophore will show activity unless we test them. So down selection of the compounds for in vitro studies first, and then go for the in vivo studies if that shows promise the in vitro assays. Then iterative 3D shape-based pharmacophore searching in conjunction with large compound databases have the efficacy of finding, or have the potential to find very good compounds very quickly. And the in silico ADME toxicity evaluations and docking methods are quite useful for down selection. So in short, we can say the in silico methods can maximize the efficiency of the expensive therapeutic discovery and development process. And finally, I think the one more slide shows, some acknowledgements. This is the non-oxime thing with our colleagues in Walter Reed, and this is the tryptantane also with our colleagues in Walter Reed. I hope that's it. And these are some of the references. And that's it. Thank you for your attention. And if you have any questions, you can please let me know if I can answer them right now, or you can send me. I'll try to answer all of them.
So there is one question in chat box.
Oh, okay. So how do I go? Okay, I can go to the chat box through, okay, I can shorten this, I guess.
Sir, it will be better if Tanushri ma'am.
Okay, I'm just saying you the question. Is
Sure.
there any adverse or favorable effect of pharmacophore on animal body fluid, which is mostly H2O?
No. You see, pharmacophore is going to discover, or rather identify new compounds. Okay, the idea is that since, if you go back to my first slide, that this is a very expensive process of drug discovery. Now, why in silico methods are necessary and virtual screening is necessary is because it can shorten the, I would say that shorten the length of the process and at the same time the efficacy of the process by down selecting the compounds. Now, the idea is that if we get a new compound, we'll have to test that in vitro, first in vitro, and then it's necessary to go for the in vivo test. So, you know, we don't have to, Pharmacophore is not going inside the thing. Pharmacophore is giving you an abstract thought about the molecular, intrinsic molecular reactivity. See, the molecule is carrying certain active nature. Now, whether it will work in the body, that has to be proven by experiments. See, in silico means, you know, only in the computer. Unless we really perform the experiments, we are not certain about this activity. And when it reacts in the body, there are many functions of the body. You know, the molecule may just be unavailable to the body. Like, you know, I mentioned about the tryptans. They are like hard rocks. They don't go. They are not, you know, bioavailable. That means even if it goes there, it is not available to the body. So that is why it may not function. The water is there. Water, the protein structure, all of them are there. You are right. But it has to circumvent these problems in the body. And this can be calculated even by molecular dynamics to get very close to the protein structure and see what are the functions of the water molecules at that time. I hope I could answer some of the...
Thank you, sir. Excellent, excellent, sir. Thank you very much. Thank you very much.
Sir, excuse me, sir.
Yes, please.
May I ask one question, sir, if you permit?
Yeah, sure. Why not? I'm here to...
Okay. Myself, Deva Prasad Ghosh from Kite School of Pharmacy.
Okay.
Sir, my research is on pharmacy, sir. As you know that you are talking about this molecule having some difficulty in its bioavailability. So is that due to solubility or is that due to enzymatic degradation?
You know, as far as we know, it is the solubility issue was there. So what we did at that time, that we took it in DMSO.
Okay.
DMSO as a solvent, we use that to inject in the body of the mice and to see that whether it can go to the target to really work against the malaria strain. But unfortunately, it doesn't go. It goes there, but you know, it is eliminated by the system very quickly, because you know the molecule has to be, if it is an oral compound, if in the intramuscular injection and other things you provide, then obviously it goes to the blood and then goes to the system in different ways to absorb and you know function as a drug, right? Now,
Fast metabolism.
Yeah. So, but you know, orally it goes to the you know stomach and then you know the intestine,
liver,
and other places, liver, and then it goes to the blood. So, but you know, pharmacophore is not going to define that kind of thing. But tryptans, you know, what we did that since various ways they tried, you know, to make it successful in the body, it never worked. All the things failed for targeting malaria by tryptans. So the army decided to discard this in the early 2000s. I mean, the entire 5-6 years this was worked there. It did not really work. So I'm not sure about this because I know I didn't pursue any further study of tryptans. But if you are doing any work on tryptans, I can share some of our knowledge on this one.
So actually, I'm thinking, so from where I can obtain this molecule, sir? Is it commercially available?
Actually, it is. This is the Strovithensis QCA. This is a plant which is probably available in India also. I don't know. You'll have to contact some of the natural product companies whether they can help you in extracting the tryptans. It's probably possible. Yeah.
Kindly
Yeah,
share some data to so I can search.
Yeah, yeah. You can send me an email. I can attach some of the recent, I would say that Tritantin articles, because they still correspond with me in many ways. So I can forward you quite a few. You send me an email, okay?
Okay,
My
sir.
email address
Kindly
is,
give me your email ID.
I think my email address,
Email
The
ID is
email
provided,
has
sir.
been shared in chat box.
very first slide, I had the email address.
Yes, sir. Yes. Okay. Okay, sir. I will take it from the chat box. Thank you. Thank you.
You're welcome. Or if you
So-
want, yeah, I
Sir,
can
I-
write down my email address. What is your name? Here.
Sir, Deva Prasad Ghosh.
Devo Prasad Ghosh. Devo Prasad Ghosh. Yeah, I got it.
Sir,
Oh.
sir, sir,
Oh, I cannot. I think I cannot write it here. Can I?
Sir, actually, your email ID has been written in the chat box by our organizers.
Oh, okay.
They
Okay, you can take it from there. Okay.
স্যার
Is there any other? Sure.
স্যার আমি স্যার বাংলাতেই বলছি স্যার
Sure.
স্যার অ্যাকচুয়ালি এই প্যান্ডেমিকের সময় আমরা দেখেছি মানে অ্যাকচুয়ালি আমরা জানলাম যে স্যার হিউম্যান ট্রায়ালে অ্যাকচুয়ালি 2 to 3 ইয়ার্স অলমোস্ট টাইম লাগে যে কোনো একটা মেডিসিন বেরোতে কিন্তু স্যার এই কোভিড এর পর আমরা দেখলাম অলমোস্ট 1 ইয়ারের মধ্যে ভ্যাকসিন বেরিয়ে গেছে এবং ইভেন সেটা গভর্নমেন্ট থেকে এ করে ইমিউনাইজেশন প্রোগ্রামও শুরু হয়েছে। সো স্যার ভ্যাকসিনটার কতটা কাজে লাগবে সেটা মানে রিলায়েবিলিটি না শুধু স্যার মার্কেটিং এতেই অ্যাকচুয়ালি স্টার্ট করে দেওয়া হলো, সেটা হিউম্যানের উপরে কি অ্যাডভার্স এফেক্ট পড়বে কিনা স্যার সেটাই।
দেখো ভাই, দিস ইজ পার্টিকুলারলি আমাদের এই দেশে দুটো ভ্যাকসিন ফাইজার অ্যান্ড মডার্না, এরা দুটো ভ্যাকসিন বের করেছে, কিন্তু এটা হাইলি মানে আই উড সে দ্যাট সায়েন্টিফিক্যালি খুবই সাউন্ড। এটা ওরা প্রথমে তোমার জেনেটিক ইয়েটা নিয়ে ভাইরাসের ওরা আইডেন্টিফাই করেছে হুইচ আর দ্য জিনস হ্যাভ অ্যাসোসিয়েটেড উইথ দ্যাট, তারপরে প্রোটিনটাকে আইসোলেট করেছে ওরা অ্যাবসলিউটলি দ্য স্পাইক প্রোটিনটাকে নিয়ে তারপরে ওটাকে মিমিক করে একটা আরএনএ ডিজাইন করেছে এবং দ্যাট আরএনএ হ্যাজ বিন ইনজেক্টেড এবং দে হ্যাভ ফাউন্ড 30,000 পিপল হিয়ার ইন দ্য ফার্স্ট ট্রায়ালে, তারপরে সেকেন্ড ট্রায়ালও করেছে, আরো অনেক বেশি মানুষকে দিয়েছে এবং দে আর অলওয়েজ সাকসেসফুল। সো আমার মানে এই ভ্যাকসিনটার উপরে খুব স্ট্রং বিলিভ আছে। তবে এটা ফ্যাক্ট যে এটা এমার্জেন্সি অ্যাপ্রুভাল হয়েছে, ইট ইজ নট এ পার্মানেন্ট অ্যাপ্রুভাল। এখন তো দেখবে 10 বছরের ভেতরে কোনো এফেক্ট হয় কিনা, তারপরে এফডিএ এটাকে ফাইনালি অ্যাপ্রুভ করবে। নর্মালি এই সিচুয়েশনে তুমি মানে ইউ ক্যান ওয়েল আন্ডারস্ট্যান্ড দ্যাট অ্যাবাউট 400,000 people have died already, 413,000 people, and every day in this country 4,000 people on the average dying. এটা ওটা নাকি যেন নট এ অর্ডিনারি সিচুয়েশন না, এটা হচ্ছে ভেরি সিরিয়াস এন্ড ভেরি গ্রেভ সিচুয়েশন। তো এখানে যদি একটা ভ্যাকসিনের সাকসেস দেখা গেছে ইন হিউম্যান ট্রায়ালস উইথ দ্য 30,000 ভলেন্টিয়ার্স ইন দ্য ফার্স্ট ফেজ, সেকেন্ড ফেজে আরো বেশি মানুষকে, আমি এক্স্যাক্টলি নাম্বারটা বলতে পারছি না, বাট সো ওয়াই নট দ্যাট শুড বি ইউটিলাইজ। এবং পৃথিবীর অন্যান্য দেশেও এই একই প্রসেসে ওরা আরএনএ ভ্যাকসিন তৈরি করেছে। আমি জানি না ইয়েতে বোধহয় অ্যাস্ট্রোজেনিকে বলে যে কোম্পানিটা অক্সফোর্ডের, ওরা বোধহয় একটু অন্যরকম ওয়েতে তৈরি করেছে, অ্যাডজুভ্যান্ট দিয়ে এবং কিছু দিয়ে একটা ডেটস ভাইরাসকে নিয়ে করেছে। আমি জানি ইন্ডিয়াতে কিরকম করে করেছে, একটা কোম্পানি ইন্ডিয়াতে খুব নাম করেছে, নামটা কি ভুলে গেলাম, ভারত বায়োটেক। ওরা কি করেছে কোন এক্স্যাক্টলি প্রসেসটা আমি জানি না, কিন্তু ইয়েতে অক্সফোর্ড অ্যাস্ট্রোজেনিকা যেটা সিরাম ইনস্টিটিউটে বোধহয় তৈরি করছে, সিরাম ইনস্টিটিউট তো পুনাতে, তাই না? হ্যাঁ, ওখানে যেটা তৈরি করছে দে আর ডুইং ফলোইং দ্য সেম কাইন্ড অফ মেকানিজম, অনলি ডিফারেন্স ইজ দ্যাট ওরা ডাইরেক্টলি মিমিক করে যে সিনথেটিক্যালি আরএনএ এটা তৈরি করেনি, ওরা ভাইরাসটাকে ডেড ভাইরাসটাকে নিয়ে ওরা ওয়ার্ক করেছে ইন দ্য ভ্যাকসিন as far as I understand. কিন্তু আমার মনে হয় যেন this is under this terrible situation, these vaccines are very useful and very sound to ইয়ে আমাকে এখনো দেয়নি, আদারওয়াইজ আই উড হ্যাভ টেকেন ইট। কারণ এটা প্র্যাকটিক্যালি ঘরে বসে থাকতে বাধ্য হচ্ছি বিকজ অফ দি। তবে নিউ ভ্যারিয়েন্টে কিরকম ওয়ার্ক করবে এখনো সবাই জানে না, তবে বলছে তো ওটা নাকি যে নিউ ভ্যারিয়েন্ট যেটা ইউকে থেকে এসছে, দ্যাট শুড বি ওয়ার্কিং। I mean the vaccine should work against that also.
স্যার অ্যাকচুয়ালি আমাদের ইন্ডিয়াতে অ্যাকচুয়ালি ফেসবুকে মানে সোশ্যাল মিডিয়াতে একটা মিমিক চলে, যেটা হচ্ছে স্যার ভ্যাকসিনের সাকসেস রেট অ্যারাউন্ড 95%। আর আমাদের ইন্ডিয়া উইদাউট ভ্যাকসিন অ্যাকচুয়ালি যখন ভ্যাকসিন বেরোয়নি তখন আমাদের মানে রিকভারি রেট ছিল অ্যারাউন্ড 94%। সো কোশ্চেন মানে মানে অ্যাকচুয়ালি সোশ্যাল মিডিয়াতে লোকেরা মিমিক করে যে আমরা তাহলে ভ্যাকসিনটা নেব কেন, এমনি যদি।
তবে এটা ঠিক মানে আমি জানি না, অনেক ক্ষেত্রে ধরো ইমিউনিটি ইন্ডিয়ানসদের বেশি থাকে, হ্যাঁ ওটা অনেকেই বলে। হ্যাঁ আমরা ছোটবেলায় ইন্ডিয়াতে we have taken BCG and various other vaccines, so probably they help, I'm not sure. কিন্তু ভ্যাকসিনটা নিলে you are secure, very little chance that you get infected by COVID. তবে প্রশ্ন হচ্ছে যে কোয়ালিটি অফ দ্য ভ্যাকসিন যদি মানে মানুষকে মেরে ফেলে, then of course it cannot be taken. But I think there is no such report so far. I don't know anybody in any country হ্যাঁ ভ্যাকসিন নেওয়ার পরে এই COVID-19 ভ্যাকসিনের পরে মারা গেছে।
কিছু রিপোর্ট এখনো
হয়তো কিছু অ্যালার্জিক রিয়াকশন হয়েছে, আমাদের এই দেশেও হয়েছে, হ্যাঁ কিন্তু অ্যালার্জি এবং অনেক ক্ষেত্রে এই জন্য ডাক্তার পার্সোনাল ডাক্তারের প্রেজেন্সে যদি ভ্যাকসিনটা নেওয়া যায়, I don't think it will be harmful. It will rather help you in the long run. আমার মনে হয়।
Yes. Thank you, sir.
Sir, I have already, sir, I have mailed you, sir. Sir, I have mailed you regarding that molecule, anti-malarial molecule.
Okay, I'll mail it back, no problem.
Thank
Okay.
you.
Anybody has any other
I cannot hear you properly. Hello? Hello?
Hello.
Yes?
You are not audible. Sir, we can now end the session, sir.
Oh, you'll be ending the session. Very good. Thank you very much for your all the attention. But you know, if you have any further questions, you can send me in an email. I will try to answer them all. Okay?
Thank you, sir. Thank you, sir.
Thank you.
You're welcome, Dr.
Thank you, sir.
Mitro, thank you for giving me this opportunity. And I really enjoyed this talking to you because I talk little more probably.
Okay. No matter, sir. Thank you, sir.
Thank you. You're welcome. Take care.
Okay, sir.
So should I log out? Okay.
Yes, sir.
Thank you.
Yes.
Thank you very much. Bye.
Bye, sir.
Bye.
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 You can send me any questions if you want. Later.
Okay.
Ashish sir, my voice is audible now?
Now
What?
it is clear. Now it is clear for Mr. Man.
Okay, so I'm just not putting my camera on. So our next speaker is Dr. Subhabrata Majumdar. He's from New York, USA. He will be talking on the topic finding needles in a haystack or determining key molecular descriptors associated with the blood-brain barrier entry of chemical compounds using machine learning. Dr. Subhabrata Majumdar is a senior inventive scientist in the data science and AI research group of AT&T Lab. His research interests have two focus areas: statistical machine learning, specifically predictive modeling and complex high-dimensional inference, and secondly, trustworthy machine learning methods with emphasis on human-centric qualities such as social good, robustness, fairness, privacy protection, and causality. Dr. Majumdar has a PhD in statistics from the School of Statistics, University of Minnesota, under the guidance of Professor Anshu Chatterjee. His thesis was on developing inferential method based on statistical depth functions focusing on robust dimension reduction and variable selection. Before joining AT&T lab, he was a postdoctoral research at the University of Florida Informatics Institute under Professor George Michael Ledis, and he has an extensive experience in applied statistical research with past and present collaborations spanning diverse areas like statistical chemistry, public health, behavioral genetics, and climate science. He recently co-founded the Trustworthy ML initiative to bring together the community of researchers and practitioners working in that field and lower barriers to entry for newcomers. Sir, I will request you to please take over the session.
Just checking, can you hear me and see the slides?
Yes, sir, you are audible and your camera is also okay.
Okay,
Yes,
the
sir.
slides are on also, right?
And
Yes,
slides are also visible.
Great, thank you. Thanks for the introduction and thanks for the invitation for speaking in your event. And since the last talk ended a little late, I'll go a little fast and try to make up for the lost time. So as mentioned in the introduction, I have interest in like a lot of aspects, applied aspects of statistics and data science. And as part of that, I've had a bunch of collaborations with Professor Subhabrata Majumdar, who a lot of you might have heard in his talk yesterday on kind of a similar topic. So today I'm going to speak about one of our past projects, which I thought would be interesting to this audience because of the kind of data that we work with and the kind of methods that we work with. So to give a little bit of context in why we might care about such a statistical approach in advanced materials. In material science, it's a broad area, and statistical methods, big data are getting used in this field more and more as time goes by. Computation becomes easy, people have access to more data and data generation procedures. So this happens in a lot of different types of materials, but the general theme is kind of the same, is that you want to infer some properties of these materials from their inherent structures. So essentially, the high level problem is this, that from the structures, you feed the structures of a bunch of materials, chemical compounds into a statistical model like regression. It could be as simple as that, or it could be a very complicated machine learning model. And then you want to find out that whether they perform good or bad in a certain task, like for example, killing certain types of cancer cells, anything that's practically relevant, essentially. And the reason this is important is that you can sort of bypass a lot of lab testing using this kind of computational procedures, in the sense that you don't really need to do lab tests for hundreds of compounds to find out whether they are good or bad for your purpose. What you can do is run this big set of compounds through this model to shortlist, say, 10 compounds or 15 compounds for a certain task, or precursors of the final materials for a certain task. And you do testing, actual lab testing, in vitro testing on these compounds. So that's where the value of these in silico methods are, to sort of prioritize among a large set of candidate compounds to be more efficient in terms of cost. And coming to the context of chemical compounds, drug discovery a little more, what we will be talking about is QSAR. Again, you might have heard this term from Professor Bosak's talk. The full form is quantitative structure-activity relationship. And the core of this is the relationship between the two things, structure and activity. This is what I had mentioned before, which is that you are trying to correlate the inherent structure that is there in a chemical compound. For example, the number of double bonds in the compound, number of carbon atoms in the compound. Of course, the actual descriptors are more complicated than this, but I'm just giving an example. So you want to correlate these kind of inherent structures of these chemical compounds that we can measure and observe to certain properties that are not really that you don't really know the cause-effect relationship between the compound and the function completely. There is some sort of an uncertainty involved in that activity. So you want to get a more overall view, more predictive view of the activity from the structure. You want to minimize the uncertainty in predicting that activity. And the materials that we are going to talk about in this specific session are chemical compounds that might be candidates for a certain medicine. It might be in the medicine delivery in the human body. It might be the medicine as itself. It might be precursors to some final chemical compound that is used as a medicine. But the purpose is the same, is that we want to shortlist a number of chemical compounds from a large number of compounds. And the process is exactly the same as I had mentioned before. You calculate descriptors using some softwares that are very contextual to the chemical community. A lot of people have their own in-house software, a lot of companies or universities. You may compute a bunch of chemical descriptors, the inherent property descriptors, structural descriptors, using these softwares, measure the activity of these compounds and try to fit a model on those activity and structure to find out the activity of new chemical compounds that you had not used to build your model. So the structure is like this. This is kind of the original data from which you build samples, and then you kind of see that whether the sample is good enough before you try to predict for new compounds. Or also another thing you can do is to actually try to infer from chemical knowledge that why exactly is this structure causing this activity. So there is a correlation between the structure and activity that you are finding from your model. So now you want to know from the knowledge of chemists that whether there is actually any previous knowledge that says that, okay, this is the reason this structure might be causing this activity. And that way you can kind of validate the knowledge that you're gaining from your machine learning model. Okay, my screen kind of froze in the middle, but we'll get going again. And the specific type of activity that we are concerned about is whether a compound passes the blood-brain barrier or not. Blood-brain barrier is a very important component in the human nervous system, in the sense that it lets compounds, chemical compounds, selectively pass through from the blood to the inside of the brain. And in terms of drug discovery, this is important because blood-brain barrier allows different drugs to selectively enter the brain or not enter the brain. For example, if you want some drug to affect the brain, you want it to pass the blood-brain barrier. Whereas if you want some chemical compound, some drug to not affect the brain, stay in the bloodstream, then you want it to not pass the blood-brain barrier. So in a sense that it's a mediator, and you really want to design your drug compound or design the delivery of your drug compound so that it can be targeted through the blood-brain barrier or not targeted through the blood-brain barrier. And the way you measure it is called, one way to measure this is brain-blood ratio, which we will be using as our activity variable, which is the ratio of the concentration of the chemical in brain versus the concentration in blood. So if the brain-blood ratio is higher, you say that, okay, it's very high. You can kind of see that, okay, this compound crosses the blood-brain barrier. If it's very low, you say that, okay, it doesn't. So kind of putting this into statistical or machine learning kind of framework, data, structure, activity, we have the sample, which is 415 chemical compounds. The structure are computed by a bunch of in-house softwares that we are coming to, we will discuss in a moment. And the activity is this blood-brain ratio that I talked about right now. And we set a threshold that, okay, suppose above certain value, say above 90, if the blood-brain ratio can be from 0 to 100, I'm just giving an example. Above 90, if the blood-brain ratio value is above 90, you say that, okay, let's say that this compound passes the blood-brain barrier and label it as 1, otherwise label it as 0. So this becomes a prediction problem of whether the activity is 0 or activity is 1. And what we want to see here in this specific project is that whether variable selection has any effect on prediction capability. So you can compute hundreds of descriptors, thousands of descriptors, and but that's what we want to see, that whether more is always better, or there are certain descriptors which are kind of driving these blood-brain barrier penetration in this set of compounds. And that way we can at least attempt to find out some sort of interpretation of why these predictors are causing the blood-brain barrier penetration, or what properties of these chemical compounds are causing this blood-brain barrier penetration. So that's why we want to find out the top significant predictors, essentially, which are predicting blood-brain ratio, high values of blood-brain ratio, high or low. And sort of a overall schematic of what is going on here. So there is a response variable or the activity variable. 275 of them are yes, so set as 1, others are 0. And the descriptor set, there are 2 descriptor sets, one each computed by 2 of our groups that we were collaborating with. One of us was Professor Boschak's group, and another was Claudiu Lungu and Mircea Dudea, a group from Romania. So they also had their own in-house software, and we kind of wanted to see that whether our descriptor, how do our descriptor perform by itself, theirs perform by itself, and combined together, what happens. So there were this many descriptors, so you can see that there are a lot of them. But as we'll show, there are some descriptors that will come up as high importance. And what we do is just build a model, do some validation to see that the model is performing well, and also do the variable selection that I mentioned. A little bit about this descriptors. The first set, as I mentioned, was computed by the, sorry, I think I did it the other way, but the first set was computed by the Romanian group, and that had 579 descriptors, and the second had almost 200 by Professor Boschak's team. And notice one thing, that these descriptors are very specific to the software that is being used. And so depending on that, there could be other descriptors as well, based on what exact software you're using. And the reason I want to point this out is that because of this reason, since the compound itself is sort of a finite compound, it's like it has, say, 30 atoms, and there are base pairs, X many base pairs in them, and 15 single bonds and 12 double bonds, things like that. The compound is like a finite thing. But since you can compute these descriptors in multiple ways, what happens often is that there is a lot of dependency among these descriptors. There is a lot of collinearity among these descriptors, or other types of correlation among these descriptors. And that way the prediction problem becomes harder, in the sense that if some descriptors comes up in the top, that doesn't mean that other descriptors are not always unimportant, but you really need to be sure that you can't apply any method just to fit a model. The method that we used is called random forest. It's a pretty well-known method, which essentially combines the predictions from multiple decision trees. And we use this because it has been proven effective in the QSR context before. And what it does is that it trains a bunch of decision trees based on the data set, and gets the result from each of these decision trees. For example, in our context, the result will be the prediction of the activity that is being measured. So 0-1 blood-brain ratio, whether the ratio is above a certain threshold or not. Suppose decision tree 1 predicts that the ratio is 1. The activity value is 1, thresholded ratio. Decision tree also says that it's 1. And decision tree 3, suppose there are 3 decision trees. There are n trees here, but I'm just saying n equal to 3. So first tree says result is 1, second tree says result is 0, third one says result is 1. And then we do a majority voting for this kind of descriptors. You average them if the activity is continuous, but since the activity is 0-1 here, we do majority voting. So majority voting is nothing, it's just a fancy term for saying whoever has the most votes wins. So in that case, 1 wins because decision tree 1 said that result is 1, 3 also said that result is 1. So the final result is taken as 1. So that's how the random forest predictions happen. And what you can also do is average the outputs instead, even if they're 0 and 1. That way the final result will be a probability such that the outcome is 1. So in that case, that probability is 2/3. And generally in random forest models, there are a lot more trees. So you get like a more continuous form of probability if you do choose to make the probability prediction. All right, so in terms of variable importance, we want to see that if we take the top 5% variables, whether they improve model performance, how we measure model performance that we come to later, but that's how we want to measure that. If we want to take top 5%, if they improve model performance, then the full model, which is the model of all variables, if we take the top 10%, 15%, 20%, up to 95%. And then see that, observe that whether the improvement actually happens for a small number of descriptors. And that model is the model that we would be interested in. Because using a small number of descriptors, training a model using that has a lot of computational savings. You don't need to calculate all the descriptors, you just need to get these and you're done. So that's sort of the motivation. Also, one small thing is that the variable importance depends on the training model. And since you are training on one part and testing on another part to evaluate the model, you should use a separate full model for each train test split to get the evaluation metrics, because the train test splits are random. So the evaluation metrics will also be different for different train test splits. For example, accuracy. Accuracy might be slightly different for different splits of training test of this 415 compound data set. And that's why you do different, you do a large number of splits, then average those metrics to find out a more stable value of the evaluation metric. And while doing that, this just says that you should be doing different variable selection steps for each of these splits. And the metrics, finally, area under curve, which some of you might know is basically the area covered under a receiver operating characteristic curve that plots the precision and recall values. Precision is true positive and recall is 1 - 2 negative. And top 20% lift, this essentially measures at the top of the AUC what is the accuracy. So that's useful in the QSAR situation, because often that always happens in a practical situation, that there are limited resources and you only can screen, say, 20 chemicals. So in that case, you'd want to know, you're not interested in what is the overall performance in the model, but you want to see that how do the top candidates in the model perform. You want them to perform well. You don't care about what happens elsewhere in other test compounds. You just want to make sure that the top test compounds that you're selecting, they are highly likely to succeed. They are highly likely to be actually useful for your purpose. And that's where that's what this metric measures. And also there are a bunch of other well-known metrics that we use. coming to the results, first let's check out a few of the high importance compounds, high importance descriptors. As we found out that some IC indices came up on top, which are related to the heterogeneity of atomic neighborhoods. And on the other hand, these other descriptors also came up, which are triplet descriptors, and they represent the polarity of the molecules, and which has been correlated with the blood-brain barrier entry of chemical compounds. In the blood-brain barrier entry of chemical compounds. So that was kind of reassuring in the sense that we are finding something useful that people have found out before. On the other side, for the second set of descriptors, we found out these 4 or 5 descriptors rather present in both of the combined and their own set of models. And all of these express not all of these, the first 3 at least, they express the presence of heteroatoms. And the fourth one, A log P3, is a quantifier of hydrophobicity. And both of these things that are quantified by these descriptors, polar surface area and hydrophobicity, and also the C log P3 has been found to be influential in the prediction of blood-brain barrier. So that's also an indication that there is a heterogeneity among the importance of the descriptors in this data at least. So that sort of gave us an indication that descriptor selection might do something useful because previous knowledge is replicated to a certain extent by our outcomes. And after the validation, we are kind of sure that, okay, the performance increase is actually happening. So as you can see here, the area under curve, if it's good, it's here. If it's good, it has higher value. Same for the lift, sensitivity, and specificity. If the model is performing well, it has higher value. And you can see that if we use 0% predictor, that's low, that's expected. But something useful happens when you use like 5% or 10% predictors for at least the DBEA and combined set of descriptors, in the sense that they actually perform better than the full model. So as you can see, the performance is the highest at 5%, and then actually goes down slightly to become like 0.82 at 100% predictors when we use 100% predictors to build a model. So that was our full model, the original model. But instead, if we just use the top 5% predictors in the original model, build another model that actually performs better than the original model. And this performance guarantee, or rather this performance, holds for all the metrics. You can see that for AUC, this holds. For 20% lift, it holds. For specificity, it holds. And for sensitivity, it's about the same. But the advantage is quite visible for the other metrics. And also there were other 2 as well, which I'm not showing here right now because the shape of the slide, but it happens for the others also. So there is an indication that variable selection is working. But as we can see, that doesn't happen for the first set of descriptors. And that is kind of expected in the sense that not all descriptors might be useful. And for this specific compounds, the one set of descriptors works well. But the takeaway from here is that variable selection approach can give a better model. All right, so finally, to wrap it up, we demonstrate the importance of parsimonious model, which is a model with less number of descriptors. So sometimes less is more. However, we do need to select descriptors that are most effective. Also, another thing is that variable selection is quite general technique. It's applied everywhere whenever people work with data. So can be applied to other problems that involve statistics or data science in material science, and also how exactly variable selection is effective and what contexts in what kind of problems, and also how do the resulting models compare with full models in terms of things like applicability, domain, and others. That's also needed. So with that, I'll finish. Sorry, I came out of the one. Yeah. All right. Thank you so much, and open for questions, if any.
Sir? Hello?
Yeah.
Sir, Devaprasad Ghosh from High School of Pharmacy.
Right.
Sir, so have you taken this lipophilicity, hydrophilicity, particles, everything under consideration while deciding, setting these variables?
Mm-hmm. So I think, as I mentioned here, let's see. Yeah, so the C-log-P measures hydrophilicity, or rather hydrophobicity, so less value is hydrophilicity. So in that sense, all of these high-level properties are measured by a combination of these variables. And that's actually a good point in terms of demonstrating whatever I was talking about, which is that a lot of these features can be correlated. So for example, C-log-P measures hydrophobicity in this set of descriptors. But it might as well happen that in this set of descriptors, some other combination of features combinedly also measure hydrophilicity. So in that case, there will be some correlation within these descriptors.
Then how particle size, will particle size also be important? Because sir, we know if it is less than 500 Dalton, they can easily pass any barrier.
Mm-hmm. Yeah, I would assume so. And in that case, you'd really want to map those particle size and those kind of properties to these features. See that what features would be useful in measuring those properties, and then go back to the model and see that where are those features, how much importance do these features have.
Okay, sir. Thank you, sir. Thank you.
Yeah.
So till now we don't have any other questions, but we have shared your email ID.
Yeah, great.
So if they have any query, they can just email you.
All right, no problem. Yeah.
Yes. Thank you, sir.
Mm-hmm.
Thank you, sir, for your wonderful lecture. So we will end our session here. And thank you, sir, once again. So for participants, tomorrow's session, again, it will start from 10:30 a.m. So everyone, please join before time.
হ্যাঁ এন্ড করে দিতে তুমি করে দিচ্ছ না আমি করে দেবো
আমি করে দিচ্ছি।
করে